Hi, so welcome to Tuesday afternoon, and it is A-level biology time, and today we are going through the AS paper, the OCR AS paper from 2017, and in particular, Paper 1. So I am going to go through them. I'm going to show you what I would have written as my answers. I'm going to show you where I would have got the marks and also where I wouldn't have got the marks. Get them all. And I'm going to hopefully help you understand and decipher all the different things that you need to be thinking about while you're doing an exam paper. I don't write exactly what is in the mark scheme, but I am going to show you exactly where I got the marks. So I do that by using ticks with mark points 1, 2, 3, and those correspond to the mark points that are in the mark scheme. Hopefully that will become clear as we go along. And I put these ticks exactly where I think, next to the word, where I think I've absolutely nailed the mark. So without any further ado, what I should also say is that if you're not in live stream land, what you should be doing is stopping the video as we come to a question and having a go at it yourself, seeing what you would have written, and then have a look at what I would have written. I'll then show you the mark scheme on the left-hand side of the screen. I'll show you the examiner's comments about marking on the right-hand side of the screen, and then I'll show you what I would have got. So let's get going. First of all, as with OCR papers, you start off with a whole load of multiple choice questions. So here we have a diagram of the epithelial lining of the trachea. And hopefully you'll recognise that these long columnar cells, because they're like columns, have got cilia on the top. So these are the ciliated epithelial cells, cell Y. Cell X is a goblet cell which is secreting mucus. And of course, the mucus is going to capture the pathogens, and the cilia are going to mucus and the pathogens up the trachea so it can be swallowed. Anyway, back to the question. What are the following statements about tracheal, what following statements about tracheal cells are correct? These are all columnar epithelial cells. They're absolutely not, because these ones look nothing like columns, and neither does the cell, let me say, cell Z and cell X are not columnar. So that is not correct. Cells X and Y move mucus and track bacteria out of the trachea. Well, that is not true, because it's only cell Y that does the moving, because they are the ciliated cells. Cell X releases mucus into the trachea. Is true. Cell Z is a goblet cell, and that is not true, because cell X is. So the answer is cell C, and here are the answers to the multiple choice, and that is correct. Okay, DNA is formed from 3 main groups of molecules: pentose sugars, phosphate groups, and nitrogenous bases. The bases are split into purines and pyrimidines. Identify the 2 purines. Now, the way that I remember what purines are, now I know this is silly, I remember that they're A and G, and that is the chemical symbol for silver. And I just think of that being pure. In other words, a purine. That's the way that I remember it. And so I'm now looking for adenine and guanine. And the first one that I come across is A, so that is that answer. Which of the options A to D is the reason why plants require specialised transport tissue? Why do they need it? To allow space? No, that isn't why they have transport tissue. Because they all have a large surface area to volume ratio. Well, that is not true, because plants have a low surface area to volume ratio, which is why they need the transport tissue. So if only this said small, I would have said that was right, but it's not, because it says large. To carry, I can't see what that is, something to their leaves. I can't, solutes to the leaves, it's on my screen, so I'm sorry, I can't read it. And And I think D says to overcome, oh, I now can't remember. Hold on, let's see. To overcome the limitations of diffusion over large distances. Well, this is the correct answer. They don't have transport tissue only to carry, is it solutes? Sucrose to the leaves. Well, sucrose isn't carried to the leaves, it's transported from the leaves. That is incorrect. So the only right answer is D. And here is the mark scheme. Question 4. Genetic diversity of a population can be estimated using the following formula. And you've got it here. And hopefully that is familiar to you because it is part of your syllabus. In 1992, a study estimated the genetic diversity of 4 isolated populations of lions. They recorded the number of gene variants at a selection of gene loci in each population. Which of the populations of lion has the greatest proportion of polymorphic gene loci? In other words, if you divided this number by this number, what is going to be the biggest number? So this is actually a maths question. So I'm immediately looking, I'm scanning down these numbers and I'm going 73 out of 1927 is much less than 1,110 out of 2,156. So this is definitely not it. And I'm now looking at these numbers and I'm getting my calculator out at this point because I'm using a little bit of mental math. And I'm spotting that this would equal exactly 0.5. I'm now looking at this one, 1,030 out of, well, this is a smaller number out of a bigger number. This is a bigger number and this is a smaller number. So I am thinking, without even using a calculator, I'm thinking that it's B. Now, I think what I did do, in all honesty, is that I out the maths for B and C because I could see that both of these was over 0.5. So I was down to only actually plugging 2 sets of figures into my calculator, but already I'm pretty sure that it's B. Hope that kind of logic makes sense. Many plants are adapted to the availability of water in their environment. One group of these plants are the xerophytes. Which of the following statements correctly describes a xerophyte? Smooth cord grass in highly saline environment. No, I would call that an extremophile, something in a very extreme environment. So don't think that's right. Water lily has parenchyma tissue. Don't even know what that is. Don't really care. To allow the movement of gases to submerge roots. Okay. Yeah, I don't think, I'm not convinced about that. Don't really, yeah. Oh, sorry, water lily. Water lily, here's the thing. This is something that lives in water. A xerophyte is a plant that lives in very water deprived areas. C, a poison tree that has leafless branches covered in thorns to leafless branches covered in thorns. So in other words, the thorns have replaced the leaves to reduce water loss. Oh yes, I think that's right. Water lobelia completes its entire life cycle submerged in a shallow pond. Well, to be quite frank, there it's submerged in a shallow pond. It is not a xerophyte. So it's C. This question was about you understanding what a xerophyte is and being able to identify a description of a xerophyte. 6. A standard method can be used to extract DNA from the nuclei of cells in kiwi fruit. The statements below list some of the steps involved in this method. Which statement is not correct? Now, I'm going to tell you, right, let's go through these statements first of all. A, chop the kiwi fruit to break open cell membranes. Well, as a teacher, I'm kind of going, well, what we'd normally do here is we would homogenize the kiwi fruit, or we'd blend the kiwi fruit in a kitchen blender to break open the cells. But that's kind of just mechanical chopping. So that sounds okay to me. B, add detergent to dissolve nuclear membranes. Well, that sounds okay to me. C, add protease to digest histone proteins. That definitely sounds okay to me. D, pour ice cold ethanol onto the filtrate to precipitate the DNA. And that one sounds okay to me too. I'm most sure of B, C, and D. However, chopping, you'd kind of go, that sounds absolutely. Now, I'm going to choose, I chose A, but if I'm really honest, I did not see really what was inherently wrong with chopping, because who's to say that the person didn't chop the flipping kiwi into a mush, which is basically what happens when you blend something. Now, this is a question. I looked at the examiner's report for this question, and very few students got the right answer. And the examiner's report said that every answer, every wrong answer, got an equal number of students chose each of the wrong answers. In other words, students were really confused by this question. And I'm going to be honest, I am not in the least bit surprised. So this is a case of, if you know your biology, if you know your stuff, and you get a question like this, you know, and it's kind of stating the obvious, but go for the statement that sounds the least accurate. The best I could give you. It wasn't a question that many students got right, and I hope that makes you feel better about it. Question 7. Water, universal solvent, has the ability to dissolve many ionic and covalent compounds due to the water's polar nature. Which of the 3 carbon compounds below will not form hydrogen bonds, therefore not dissolve in water? I am looking for compounds that do not have the atoms oxygen or nitrogen in them, because those 2 atoms are those really electron greedy atoms, and therefore they would make the molecule a polar molecule, because here, for example, this would become slightly negative and this hydrogen would become slightly positive, or maybe this hydrogen would become slightly, because the oxygen would be holding, it would take an unfair share of the electrons. There would be an unequal distribution of charge across the molecule, and that is the definition of a polar molecule. So the only molecule that doesn't have oxygen or nitrogen in it is the propane. So it's D. And here is the mark scheme. Oh, Lavinia, just to say hello. Thank you for your little greeting there. Question 8, which of the options A to D is a correct statement about polysaccharides of glucose? So this is about reading the carefully and making sure you know your stuff, knowing the detail. Cellulose microfibrils are formed by hydrogen bonding between adjacent chains of alpha glucose molecules bonded with 1,4-glycosidic bonds. I'm liking all of that, except for this bit here, which should say beta glucose molecules. So not correct. Amylose. Amylose is that helical polysaccharide part, one of the molecules that makes up. Amylose is a straight chain of alpha glucose monomers bound by 1,6-glycosidic bonds to allow for dense packing. Well, the bit that I really don't like here is that should say 1,4-glycosidic bonds. And I wouldn't really call it a straight chain. It's a single linear molecule, but it becomes a helix. Glycogen has a high proportion of 1,6-glycosidic bonds to produce a highly branched molecule for rapid release of alpha glucose. Absolutely true, yeah. D, amylopectin, this is the other starch molecule, which is a little bit branched, but not too much. Amylopectin is a mixture of 1,4-glycosidic and 1,6-glycosidic bonds between beta glucose molecules for rapid release of energy. Well, this is the bit that isn't correct at all. So it is C. And yeah. Which of the options A to D is a correct statement about tissue fluid? Carries carbon dioxide to muscle cells? No, not true, because it carries it from muscle cells. B, oncotic pressure in the capillary causes tissue fluid formation from plasma. This is not what causes tissue fluid formation. It is hydrostatic pressure that causes the formation of the tissue fluid. Oncotic pressure causes the water in the tissue fluid to move back into the capillary venous end of the capillary so that the blood volume is kept the same. So this is incorrect. Hydrostatic pressure in the capillary causes tissue fluid formation from plasma. Oh yes, that's correct. Tissue fluid is reabsorbed into the capillary by active transport. Absolutely not. This is osmosis. So the answer is C. Sorry, just having a moment there. And here is the mark scheme. Question 10, a group of students were given a 1% of an unknown digestive enzyme. They were also given three tubes containing an identical mixture of food. So here is my silly little diagram. You've got three tubes, each of which contain carbohydrates, proteins, and lipids. In each of those three tubes, the unknown enzyme is added, and they do food tests. They do a single food test. They do a biuret tube 1, an iodine food test on tube 2, and a Benedict's test on tube 3. And you're looking at the color change to work out what the unknown enzyme is. So here, this requires you to know that the iodine test tests for starch, and this is the test that changed. It went from blue-black to yellow-orange because, of course, the unknown enzyme, digestive enzyme, digested the starch into sugar. And therefore, the starch test would be positive at the beginning before adding the enzyme, and it would be negative afterwards because, of course, the starch wouldn't be there because the enzyme digested it. And that tells you that the enzyme would be a carbohydrase because starch is a carbohydrate. It's one of the complex carbohydrates, a polysaccharide. So the answer is B. DNA carries the genetic code which is non-overlapping and degenerate. Which of the options A to D contains the correct definitions of non-overlapping and degenerate? So this is A01 question asking you whether you know the definition for non-overlapping and whether you know the definition for degenerate, the degenerate nature of the genetic code. So each nucleotide, is only one part of let me say that again. A, each nucleotide is only one part of one triplet of bases. Each nucleotide is only part of one triplet of bases and the molecule breaks down easily. Well, this is the correct definition for overlapping, but this is not the correct definition for degenerate. Wrong. Follow straight after each other and the molecule breaks easily. Well, this is not the definition for overlapping and this is not the definition for degenerate. Each nucleotide is only part of one triplet of bases and more than one triplet codes for a specific amino acid. That is the definition of overlapping and this is the definition of degenerate. And finally, D, the genes follow straight after each other and more than one triplet code for codes for one specific amino acid. So this isn't the correct definition of overlapping, but this is the correct definition of degenerate. So the answer is C. And here's the scheme. Moving on, question 12. In the graph below, the top electrocardiogram shows normal heart trace, and this one below is an abnormal heart trace. What does the abnormal heart trace show? So, fibrillation, I basically think of fibrillation as where this heartbeat trace has gone all wrong. Tachycardia is when you have more of them. An ectopic heartbeat is when you have extras of them, and bradycardia is when you have less of them. So out of these four, I would have chosen, or I did choose fibrillation, because this is when the heart, each individual heartbeat trace is basically going all over the place. 13, tropical rainforests have a very high biodiversity of plant species. Which of the statements A to D, and here's the important word, is an economic benefit of high biodiversity? Not a biological or a nature benefit, an economic, to do with money. A, high plant biodiversity decreases the animal biodiversity in the rainforest. Absolutely true, but not an economic benefit. High plant biodiversity, sorry, that's wrong, it doesn't decrease, it increases. High plant biodiversity increases the organic matter in rainforest soils. Yeah, almost certainly true, because if you've got high plant biodiversity, when they decompose, lots of different nutrients going into, or lots of organic matter going into the soil. So, but not an economic benefit. High plant biodiversity supports drug discovery and development. Oh my goodness, there is a whole industry built on this, the pharmaceutical industry. Absolutely an economic benefit. And high plant biodiversity protects the ecosystem from environmental changes. True, but not an economic benefit. So the answer is C. Now, here is the mark scheme. Worth noting here that they allowed B because there were many more heartbeats than there are in the normal trace. So worth noting that they allowed B as an answer here. 14. This is another question that caused an awful lot of problems in the year that this was set, and very few students got it right. So don't be worried if you struggle with it. Lupus is an autoimmune disease. One symptom is a facial rash, typically in a butterfly shape across the cheeks. Following a blood test, which of the following would indicate that the patient has lupus? You've had this whole description of lupus, but what we need to recognize that if there is you are trying to diagnose the specific illness, the specific infection, sorry, no, let me start that again. You're trying to diagnose the specific disease, lupus. Now it's autoimmune. It doesn't involve a pathogen. So anything that talks about, and my logic is that you would be looking for antibodies for something. But notice that you're not looking for antibodies for a pathogen, because this is an autoimmune disease. It's a disease where the body is attacking itself. So, A, the presence of antibodies for the cell surface antigens of connective tissue. Sounds a bit weird, I'm going to be frank. But I'm not going to discount it at the moment. Let's have a look at the others. The presence of herpes antibodies, well, that can't be true, because we are talking about, that implies the infection by the herpes, the pathogen, the viral pathogen that causes herpes. So I'm thinking that B isn't true. The presence of high levels of antihistamines. Now, antihistamines are a non-specific response to infection or damage. We are looking for a specific diagnosis of lupus. This can't be correct. And D, the absence of B lymphocytes. Well, that can't be true. There's nothing in the description that says that lupus wipes out B lymphocytes. If it did, the person would be in very, very serious trouble. They'd have no immunity, no specific immune response. So I don't think it's this. So I'm coming back to A. And notice that actually this makes more sense the more you're thinking about it, because we've got the presence of presence of antibodies for the cell surface antigens of the connective tissue of the person who is suffering the disease, because it's an autoimmune disease. This question, now the examiners put this down to the fact that, you know, it was a tricky application question. But I'm also going to say that it's also a very tricky question because they gave you a reasonable amount of a little bit of spurious information. This word here is almost the most important word. It's an autoimmune disease. And you needed to have a blood test that absolutely diagnosed specifically the disease lupus. So it had to be something that showed the body attacking its own tissues. Don't really care what type of tissue it is. We've got connective tissue under our skin. So, you know, not a massive jump to kind of say, oh, well, if something goes wrong, that might cause, you know, inflammation, therefore redness, a rash in that area. So I went for A, but really tricky question. 15, biologists use both phylogeny and classification to understand how different species are related. Which of the options is a statement about phylogeny? This is about you understanding the difference between phylogeny and classification. Phylogeny is where we can see how different species are related, how closely related species are. Classification is about putting species into groups. So, A, 21 species of ladybird in the UK that belong to the subfamily, whatever this is, Coccinellidae, whatever. No, that's about classification. That's about putting stuff in groups. Homo sapiens and Pan bonobo share a common ancestor. Oh, this is about relationships. I'm liking this. The lily family, Liliaceae, consists of 15 genera. Well, whoopee-doo, that's about classification. The great white shark is a member of the class, whatever it is, chondric, whatever these. No, that's about classification. So B is the right answer. And there's the mark scheme. 16, an individual bitten by a rabid dog can be treated by an injection of human rabies antibodies. What type of immunity is this treatment? Well, it hasn't been caused as a result of the person's own immune system being triggered, so therefore it is passive. And it is done by an injection, and therefore it is not natural, it is artificial. The only type of natural passive immunity that we most commonly come across is babies when they drink their mother's milk, because the antibodies are passed to the baby through the mother's milk. So I'm thinking that this is C. A student studied the structure of a blood vessel and found an outer layer of collagen fibers. Okay, well, that's one set of cells. A thick middle layer of smooth muscle and elastic tissue. That's another whole layer of cells. And it says it's a thick middle layer. And an innermost layer of endothelial cells. Which of the options is the type of blood vessel being described? I'm immediately going to say this is an artery, because they've used the word here, thick. Veins have an outer layer of collagen fiber. They have smooth muscle and elastic tissue, but it is a thin layer. And veins also have an innermost cell of endothelial cells. It's got to be an artery. Can I just point out, it cannot be a capillary, because the capillary is only made up of this layer, a single layer of endothelial cells. The walls of capillaries are one cell thick. So it can't be a capillary, because we've described 4 different types of tissue: collagen tissue, smooth muscle tissue, elastic tissue, and endothelial tissue. Can't be a capillary. So it is A. Which option, A to D, describes the role of cholesterol in cell surface membranes in the human body? Cholesterol binds to the phospholipid phosphate heads, increasing the packing of the membrane, therefore reducing the fluidity of the membrane. Well, that's not strictly true. You're far more likely to be taught that cholesterol binds to the fatty acids, because cholesterol is lipid soluble just like those fatty acids, and therefore it binds by hydrophobic interactions. So I don't think this one is right. Cholesterol binds to the phospholipid fatty acids tails, reducing the packing of the membrane, therefore increasing the fluidity. No, this is the wrong way round. It should be increasing and decreasing. So that's wrong. C, cholesterol absorbs ATP, preventing active transport. Rubbish, no. And then here, finally, we have cholesterol binds to the phospholipid fatty acid tail, increasing the packing of the membrane, therefore decreasing the fluidity. It is D. And here is the answer. Now, notice the examiners also allowed the answer A because they recognised that students may not have been taught at the detail of the fact that cholesterol binds to the fatty acid tails and not particularly to the phosphate heads. And of course, every other bit of the answer was the same. So there was a little bit of leeway there. What is the correct definition of the term coenzyme? So do you remember, yes, that's, sorry, I was just about to refer to something in the year 13. That's not fair. This is year 12, coenzyme, an inorganic ion that forms the center of a globular protein. No, because I know this is coenzymes are something, we'll get down to the finer definition, but they're something that helps make enzymes work. So we're not even describing an enzyme here. B, a molecule that binds to the enzyme, changing the shape of the active site, preventing an enzyme substrate complex. Well, that's not a coenzyme, that's a flipping inhibitor. So it's definitely not that because it's preventing an enzyme substrate complex forming. A non-protein organic molecule not permanently attached to the enzyme but needed to allow the enzyme to function. Yeah, I like that. And finally, a metal ion that attaches to the enzyme, changing the shape of the active site, increasing the likelihood of a reaction. This is the definition of a cofactor, not a coenzyme. So the answer is C. 20. During DNA replication, DNA polymerase can only work in one direction, from the 3' end of the template strand to the 5' end. This means that the lagging strand has small gaps left in the backbone. DNA ligase works to seal these gaps. Which of the options A to D identifies the bond form? Well, the DNA ligase is joining up the sugar phosphate backbone. It is not ever like DNA polymerase. Let me take the opportunity of emphasizing this. Neither DNA polymerase nor DNA ligase reform hydrogen bonds, those ones that are broken by the DNA helicase. So DNA ligase and DNA polymerase reform the phosphodiester bonds that make up the sugar, that put together the sugar phosphate backbone. So the answer is B. Glycosidic bond is something that holds together monosaccharides in a or in larger carbohydrates. Peptide bond does the same thing with amino acids making proteins, and it's not a hydrogen bond. So the answer is B, and there are the two final answers. So now let's get down to some nitty-gritty of your written answers. A student looked at a slide containing onion root tip cells under a light microscope in order to identify cells in different stages of mitosis. Figure 1 shows the diagram. Explain why onion root tips were used to view cells undergoing mitosis. Now this is what I put, because root tip cells are where mitosis occurs. Absolutely true. Here's the mark scheme. All right, here's the mark scheme. And I read this and I kind of went, oh yes, site of cell division and mitosis are the same thing. Now, in the examiner's report, I didn't give myself the mark. And can I just say, I couldn't think of it, because I looked at this and I thought I need two things. I could not think of a second thing as to why onion root tips are used. if I've already said, well, that's where mitosis actually occurs, I simply couldn't think of anything else to say. And I realised when I looked at the examiner's report that I could not give myself the mark for saying this is where mitosis occurs, because the reasoning was that that was the word given in the question. And they actually wanted you to show a more knowledge, basically, of what mitosis was. So I didn't get the mark because I should have used, I most likely term that I would have used was cell division, but I didn't use it. Looking at these two other mark points, I would never have thought of saying that it's meristematic tissue, because it's explaining why it's used. And I would have just wanted to say, because that's where, you know, cell division or mitosis occurred, not by then going on to say, oh yes, because it's meristematic tissue. I just would never have thought of saying that. This mark point 3 is actually a really valid and a really, I kind of kicked myself when I read it, because this was a really good reason as to why onion root tips are used and not the shoot tips, because of course you can also see mitosis in the shoot tip, but of course a shoot tip is going to have a stack load of chloroplasts in it, which could of course totally obscure the view of the chromosomes under the light microscope. So I thought this was a reasonable response, but here am I, not getting a single mark in this question. Suggest a stain that the student could have used to highlight the nuclei. Now I know, you know, I love the one methylene blue. This is what I put. Methylene blue is quite a good stain to remember, because it often gets you a mark for, you know, kind of a number of things. You obviously need to know iodine shows up starch, so that wouldn't have been the right answer. The other one, because I'm a teacher and I've done this practical several times, we use toluidine blue. So those are the answers that they were most expecting. But notice you could have actually given, you know, a whole load of different ones. But they could not accept, obviously, stains that were not nucleus or DNA specific. So, e.g., iodine solution. So, yeah. The student used an eyepiece graticule, which is shown in the figure. This is the EPG. The student calibrated the graticule before carrying out the root tip squash. He observed that 20 micrometers measured 2.35 divisions on the graticule. 2.35 divisions on the graticule. That can't be between here and, you know, 2.35. There's no way you could measure 2.35 on this scale. The only thing that could be 2.35 divisions on this graticule is going from here. Here's one division, here's two divisions, 2.3. Am I going to get this right? Because my eyes are going 3.5, somewhere around there. This is 2.35 divisions. Okay, and that equals, oh, sorry, I got that mark right, and that equals 20 micrometers. So calculate the diameter of the nucleus in cell C. So this is what I would have been doing. I would have been drawing on my exam paper 2 lines that just skim the edge of this nucleus. Now, can I just say that I couldn't do it accurately enough when making this PowerPoint. I couldn't get these lines to sit exactly where I wanted them to sit. This one is a fraction too far over this way, and this one is also a fraction too far over this way. It's the best I could do for illustration purposes. So here am I reminding myself that 2.35 divisions equals 20 micrometers. Now, when I worked out how many this was, I kind of came to the conclusion, and you'll have to look at it closely, I came to the conclusion that this was 1.4 divisions on the graticule. Now, if 2.35 divisions equals 20 micrometers, I've got less than that. I've got 1.4. So I've got to work out the fraction of 20 that is 1.4. So the fraction is 1.4 over 2.35. But it's that fraction of 20 micrometers. So here am I just reminding you of, actually junior school maths, where you kind of go, if ever you have something in your mind where it's something of something else, you're going to be using a time sign. Remember that 2 of the 2's is 2 times 2. So here am I going, this is this fraction of 20 micrometers. So therefore it's times by 20. And that equals 11.91. And absolutely remarkably, it's exactly the answer that was in the mark scheme. But look at the range that they allow, allowing a little bit of leeway on people's measurements. And of course, if you show you're working, it is possible that you will get 1 out of the 2 marks. I obviously got the 2 marks, but always try and show you're working, what your thinking is, because it could get you marks. Question 22. Many multicellular organisms need to be able to convert monosaccharides into polysaccharides and back again. Mammals convert the monosaccharide glucose into a highly branched polysaccharide called glycogen, which gets stored in liver cells. Explain why mammals store glycogen instead of glucose. So here we are. This is your structure, how the structure of glycogen is related to its function. So number 1, it is, so this is what I put. So here am I noticing that it's a 3-mark question here. So I'm thinking, right, I have got to, and notice that this is an explain why they use glycogen. So you're not just describing what glycogen is like, you're explaining why that is useful. So here we go. Here is my description. It is insoluble, and therefore it doesn't affect the cell's water potential. This is the explanation, okay? But clearly, for me to explain a property of glycogen, I've got to state the property in the first place. I hope that kind of makes sense. But here am I really emphasizing the difference between describe and explain. If they asked you just to describe glycogen, you'd finish here, it's insoluble. Or, if they'd said, state 3 properties of glycogen. Oh, right, yeah, it's insoluble. It has many terminal alpha glucoses. It's compact, So here's the description, here's the explanation. Here, because it has many terminal alpha glucose molecules, there's the description, and here am I thinking about my glycogen molecule, many branches, she says, going on and on. And here are all the terminal glucoses, yeah? For quick hydrolysis when required, so that those terminal glucose could be hydrolyzed. There's the explanation. Compact. Now, I use this whole term because I've seen this as being the description and this as being the explanation. So I wrote the full, this is me being a bit savvy about knowing what mark schemes sometimes have in them. So I wrote it out. I didn't just say compact. Here is the mark scheme. Glycogen is soluble, so it has no effect on water temperature. Oh, and here's the, oh, sorry, I've just drawn over this. Except, oh, I can't remember what ORA stands for now, I've just gone. Other relevant arguments, I think, sorry. So yes, notice here, you could not talk about surface area. I know that a lot of teachers tend to say, oh, glycogen is highly branched. It has a high surface area so that the glucose can be hydrolyzed quickly. And it is more accurate to say that there are a high number of terminal alpha glucose molecules. It's just a better way to describe it rather than talking about surface area of a molecule. Okay, so I got mark point 1 here, I got mark point 5 down here, and I got mark point 3. Now notice here you could just say compact, or lots can be stored in a small space. It kind of both means the same thing. So that was kind of your description and explanation in the same word. Part B, humans use the enzyme alpha amylase to break down polysaccharides in food for absorption into the blood. The gene for alpha amylase is found on chromosome 1. The gene is transcribed in the nucleus and translation occurs on the rough endoplasmic reticulum. Yes, because that's where the ribosomes are, of the cells in the salivary gland, because this is where alpha amylase is produced. Describe, now here's the thing about this question, read the question carefully. Describe how the molecule is prepared and secreted by cells of the salivary gland after translation has taken place. If it's after translation, you are not talking about protein synthesis at all. You're not talking about transcription. You're not talking about translation. You're talking about what happens after that polypeptide has been made. What happens to it in the cell until it gets secreted by the cells of the salivary gland. And what happens is that it is moved, it is transported from the RER to the Golgi apparatus. The Golgi apparatus modifies it, and then it is moved from the Golgi apparatus to the membrane, the cell surface membrane of the cell. Now, here am I trying to cut down what I have to write. So I'm telling the examiner that I'm going to use RER for ref endoplasmic reticulum, and here is my answer. RER buds off to form vesicles to transport the protein to the Golgi apparatus, where it is packed and modified. There's my 2 functions of the Golgi in my answer, showing that I know what the Golgi apparatus do. Golgi vesicles then bud off from the Golgi to transport the enzyme to the cell membrane, where it is secreted by exocytosis. Notice that I'm sounding as intelligent as I can, using as many terms as I can. So here is the mark scheme. Here are the notes from the examiner about marking, and this is where I got my marks. Now, you would think that I might have got mark point 1, transport vesicle from the RER, but I didn't. The transport vesicle is the name of the type of vesicle that moves between the RER and the Golgi. I didn't say that. I said that a vesicle was used to transport. Not quite the same thing. I didn't give myself the mark. The examiners might have been lenient, but I didn't. To the Golgi apparatus, so that's mark point 3, and here is mark point 2 for modification, processing, or folding. Golgi vesicles then bud off from the Golgi and it is secreted. So I didn't notice here, mark point 4 was for naming the other type of vesicle that you might have come across, which is a secretory vesicle, which is one, of course, which is carrying something to the outside of the cell so that the vesicle fuses with the cell membrane so that the contents of the vesicle are pushed out of the cell or removed from the cell. Exocytosis. Didn't use that word, so in the end I only got mark, well I didn't say only, I got mark 0.7. So I got 3 marks for the 3-mark question. 23. Fluid Mosaic model describes plasma membranes for all living organisms. How does the Fluid Mosaic model describe the structure of plasma membranes? So I've read this question carefully. How does the Fluid Mosaic model? Oh, okay. I can see how you can read this question 2 different ways. How does the Mosaic model describe the structure of plasma membranes? You could just do a description of a plasma membrane because you have learned the fluid mosaic model. A whole load of phospholipids, which make lipid bilayer, a whole load of proteins, you know, in that phospholipid bilayer. I focused on why, so I took the question a little bit on a different route, because I said, and I'm only just realizing it now why I've taken this different route, why I kind of was answering the question, why is it called the fluid mosaic model? How does the fluid mosaic model describe the structure? What is it about the fluid mosaic model that describes the structure? So here is my answer. Fluid, because the phospholipid bilayer, I've meant to write bilayer in here, phospholipids, or maybe phospholipids, let's just say phospholipids and proteins are in constant movement. They're dynamic, it's constantly moving. And it's called mosaic because the proteins are arranged randomly throughout the phospholipid bilayer. And that's why it's called the fluid mosaic model. Now, I must admit, I find this term randomly really quite funny because let's face it, mosaics are generally the least random pattern that you see on a floor, aren't they? Mosaic tiles are very prescribed in where all the different colours and everything are. So I just find that a bit funny. But anyway, here's the mark scheme. Here are the examiner's comments. So I ended up getting mark point 3 here because they allow movement, they move, and 4 because I described fluid mosaic. Notice that mark points 1, and I got mark 1 as well because I said phospholipid bilayer. Mark point 2 was if you actually describe the phospholipid bilayer. So, you know, hopefully those mark points make sense. B, plasma membranes are partially permeable, allowing some molecules to cross the membrane with relative ease. One molecule that crosses membranes easily is the steroid hormone progesterone. You should remember, here's the clue, which is produced in the ovaries from cholesterol. There's another clue. Explain why progesterone can move across membranes. So here is your clue, steroid, and it's made from cholesterol. You had to learn about 3 different types of lipid. You needed to learn about triglycerides, which are your kitchen fats and oils. You had to learn about phospholipids, which are obviously those molecules that make up cell membranes. And you also have to know about the group of steroids. All of these are lipids, and therefore by definition they are all lipid soluble. So if it's a steroid hormone, it is lipid soluble. So explain why progesterone can move across membranes. Well, that's because it is lipid soluble, and therefore if it is lipid soluble, it dissolves in other lipids, i.e. the group of steroids dissolve in that other group that is called the phospholipids, yeah? And specifically, so here am I being specific, in the fatty acid tails of the phospholipid bilayer, because it is the fatty acid tails that are the real lipidy bit of phospholipids. Remember that phosphate head is an ion, and therefore it is water soluble, it isn't lipid soluble. That's why I have the hydrophilic head and the hydrophobic tail, allowing simple diffusion to occur. Because of course, simple diffusion things that are lipid soluble can pass straight through the phospholipid bilayer of the cell surface membrane. They don't need any carrier proteins or channel proteins to move them. So here's the mark scheme. Hopefully that explanation helped. And here are the notes, and this is where I got my marks. Lipid soluble and dissolves. Notice that you could have absolutely said phospholipid bilayer to specify the fatty acid tails. Name one other molecule that can cross plasma membranes. Oxygen, carbon dioxide, water. There's the mark scheme. Now, I just want to point out here, name one other molecule. If you name more than one, notice, mark the first answer only. If additional incorrect answers are given, so if you had said carbon dioxide and sodium chloride, for example, the sodium chloride answer would have cancelled out your correct carbon dioxide answer. So you mustn't, if it says one, you are only allowed to give one answer. It's called the list principle of marking. You can't just give a whole load of answers and hope that one of them is correct. Clearly that wouldn't be fair. Okay, lots of other things that can go as well. Potassium ions are unable to move across membranes as they are charged. Well, they can move across membranes. They can't move across the phospholipid bilayer. Yeah. State how the structure of the cell's surface membrane allows potassium ions to enter or leave the cell. Well, that's because potassium ions are moved as a result of channel proteins in the cell. Oh, I got that right. So the membrane contains channel proteins. So, and there is your options, and here is also what was allowed, all fairly reasonable. Part 2, the process of active transport uses ATP to pump potassium ions through the cell surface membrane against its concentration gradient. ATP is made up of phosphate groups and 2 other molecules. What are they? So we should know. So ATP stands for adenosine triphosphate. We talked about the phosphate already. So now we have got to be able to name what makes up the adenosine part of ATP. And ATP is a molecule which is a derivative of an RNA nucleotide. Oh, just enough room. This is ribose and this is adenine. These 2 bits together make up the molecule adenosine. You need to know adenine and adenine. So the 2 answers are adenine and ribose, which you can obviously put in any order. Look, do not accept adenosine because that's not one of the other 2 molecules. And you had to get the pentose sugar. You had to name it correctly. But if you put just nitrogenous base and a pentose sugar, you would have at least got 1 mark if you didn't know exactly what they were. Group of students investigated the effect of temperature on the membranes of beetroot cells. This is the good old beetroot practical. A colorimeter was used to measure the concentration of purple betalain pigment that leaked out of the cells when they exposed the cells to different temperatures. And here are the results, or it's a summary of the results. They don't give you the raw data, they give you the mean, and they give you the standard deviation of the data. Using the student's t-test formula below, calculate the value of T between the data for 50 and 60 degrees C. Now, you don't need to learn this formula. This is something that is actually talked about in more detail in year 13. This exercise is about you being able to use an unfamiliar formula. So you've got to be very careful about understanding what each of the symbols means and what the mathematical formula tells you to do. So I have just arbitrarily assigned group A as 50 degrees and B as the 60 degrees numbers. So does it make sense that this is me substituting in the values from the table? 0.21 minus 0.44, one mean minus the other. Just got to make sure that you keep your group A's and your group B's the same the whole way through, and therefore it is 0.06 squared, and yeah, it doesn't really matter underneath here, but yes, anyway, 0.06 squared divided by 10, the number of readings, plus 0.18 squared divided by 10. Notice that you have to square this number and then divide it by 10, hasn't got a bracket around this squared, yeah? You square the standard deviation, then you divide it by the number. And of course, you've got to square root the whole lot. Now, this is what it's been partly worked out. Minus 0.23 divided by, when you do all of this, you end up with 0.06. So therefore, this is the answer. So here's the mark scheme. Here is where you could get marks for some part of your working. So those are the marks that I got. The critical value for T at the significance level of 5% with 18 degrees of freedom is 2.0. Crucial thing here, don't need to worry about the 18 degrees of freedom thing particularly, you just need to know that the critical value is 2.1. Use the value of T that you have calculated to explain whether the null hypothesis should be accepted or rejected. Here is what I wrote. Because 3.83 is greater than the critical value, the null hypothesis should be rejected. So this is, if we're talking about critical, so for AS level, if you're talking about comparing critical values, notice I'm talking about those critical values in the statistical table, not p-values, very different. If you're comparing critical values, the way that I remember it is that I say if the critical value is greater than, let me say that again. If the value that you've calculated is greater than the critical value, then you reject your null hypothesis. So I've used that rule here. Our value that has been calculated, it's greater than the critical value, so you reject the null hypothesis. And this is, I'm going to be honest, this is the standard, I would say the standard year 13 wording to explain why. And it's because the probability that the difference in due to chance is less than 5%. Here's the mark scheme. So this is a 2-mark question. So of course, this obviously depended on whether you got this calculation right. So the mark scheme here allowed for the 2 ways that you could have answered it, depending on what you got here. So I only got mark 0.1. I didn't get mark 0.2. Even though what's written is actually correct and is pretty much saying the same thing. However, the word significant is underlined. They absolutely demanded that in the answer in order to get the mark, so I couldn't get the mark. The students plotted the data onto a graph shown here. Describe, and explain. Describe is saying what something is like. Explaining is saying why or how it is like the way it is. So describe and explain the pattern of data shown on the graph as temperature increases. Here is what I wrote. Between 0 and 30 degrees C, I said that there is little increase in absorbance because the beetroot membrane has little damage. From 30 to 60, there is a significant increase in absorbance because the membrane has ruptured and the betylene is diffusing out. So just as an alternative answer, and it would have been absolutely reasonable, you could have said between 0 and 20, there was no increase in absorbance because these really are the same, yeah? And then you could have said between 30 and 40, 30 and 60, there is a significant increase. I think almost in some ways that would have been a better answer. I think that would have been a nicer way to describe the graph. But notice that because this is a 3-mark question, you're clearly wanting to describe portions of the graph and explain why they are the way they are. Here is the mark scheme. So notice there are marks for description and marks for explanation. So I did not get E1. I think I got, oh sorry, here are the examiner's marks. So I got D2. There is little change in absorbance because it is undamaged. At the higher temperature, there is a significant increase in absorbance because the membrane has ruptured, which disrupted. It's the same thing. So I ended up getting 4 marks, so I got 3. But really notice the difference between describing and explaining. And the bummer is sometimes when questions just say explain, so they explain why glycogen is the storage material, not glucose, you can't avoid describing something about glycogen so that you can then explain it. So it's almost a little bit sneaky because sometimes explaining answers demand an element of description because you've got to say what you are explaining. I hope that kind of makes sense. Okay, just a quick, I haven't got a slide for this, but just a reminder that I have a website and it is www.biologycarol.co.uk, and that is where I have all of my videos. And I sell the full complement of the A-level syllabus, the videos that explain the full complement of the syllabus, specifically, well, for OCR and AQA. Worth noting that every biology A-level biology syllabus covers so many things that are in common. And I sell the full 2-year course, so videos that cover everything. I sell that for £100 for you to subscribe to it for a year. So if you subscribe to it now, you would have it up until the end of your exams next year. £100 is about two private tuitions. The AS content only, so the one content, you can subscribe to that for £50. And worth saying that if you do subscribe, you will get a link, a specific link to you that means that if you share that link with your friends and they buy through that link, you get a 10% commission. So if £50 or £100 sounds a lot of money, and I really get that it is still a wodge of money to find, you could actually earn that back by giving it to your friends. And it's a way, here am I trying to make A-level biology accessible to all students regardless of the family income. And that is my genuine aim here. So anyway, and there's a freebie there, how to improve your grade. So if nothing else, grab that and have a look at that and hopefully learn from that. Anyway, back to the exam. In 1990, Carl Vose suggested a new top-level taxon to the current system of classification of living organisms, which he termed a domain. So you will know the kingdom, phylum, class, order, family, genus, species, that order that you've learned so far, and then flipping Carl Vose went and added another one on the top, which is a tad annoying, isn't it, to be quite frank. He used his results from studying RNA to organise organisms into 3 distinct groups. Named a cell component that appears in organisms in all 3 domains that Vose suggested. So you've got to think of something that all organisms, what component of the cell contains RNA in all organisms? Well, it can't be the nucleus, because you've got a whole set of organisms, the prokaryotes, that don't have nuclei. The only other component which is present in all cells that contains RNA is the ribosome. So this is an application question, expecting you to remember that ribosomes are made up of RNA and, they're fundamentally made up of RNA and a little bit of protein. So it is the ribosome. And notice this is the list rule again, because it says name the cell component. So if you'd said ribosome and nucleus, or ribosome comma nucleus, the nucleus would have cancelled out. Ribosome is the correct answer. One of the suggested is called eukarya, the eukaryotes. Name the other two domains. Well, the other two domains that are both prokaryotes are the archaea and the eubacteria. Eubacteria are the true bacteria, the ones that you think of when you have that, you learned that diagram at the beginning of AS. Archaea are also bacteria, but they're actually more closely related to eukaryotic cells. Blimey. So here are the answers. Notice you couldn't have just written bacterium, because that's just talking about one individual cell. We're talking about the domains, we're not talking about cell types. State two defining features of all members of the domain eukarya. So eukaryotes. The thing that I say to all students, eukaryotes contain a nucleus and they contain membrane-bound organelles. Those are the crucial things, which hopefully is shown up here. So I got mark 0.1 and I got mark 0.4. The others are obviously true, but those two seem to most obvious, the ones that I was most sure would be the right answer. Bose carried out a detailed study of RNA molecules in order to draw his conclusions, suggest two ways in which the scientific community are likely to have validated Bose's research. How would the wider scientific community have come to agree with Bose? Basically said, yeah, these conclusions are right. This makes sense. And here am I going back to basically your how science works content. How do you ensure that an experiment is valid? Well, you have to make sure that your methodology is correct. If the method that you're following isn't the right method and it isn't carried out precisely enough and well enough, don't want to use, if it isn't carried out well enough, then the data that you collect is going to be invalid. And also, you want to be able to repeat those results. To me, those are the two key things that make up the characteristic of validity. Methodology being correct and repetition. I would say for OCR, if they ask you about, how would you improve the validity of the data, I would always err on the side of let's critique the method. I would put repetition as my last answer, but here. So this, where's my answer gone? This is obviously my reveal. I didn't quite go in the right order. So repeating the research and critiquing the methodology. So I got the 2 marks. Notice that I could have said I got mark 0.2 because of this bit here. But notice, scientists publishing in peer-reviewed papers is basically going to mean methodology is critiqued. Scientific conferences where a lot of scientists come together and discuss and argue and debate new research that would have done it. And obviously the other answers are correct too. I think we're near the end now. Okay, semi-conservative replication describes the process by which DNA is replicated in all living organisms. Explain the meaning of the phrase semi-conservative replication. Well, that means that the DNA molecule that is made, the newly replicated DNA molecule, is made up of one old strand and one new strand. That is the definition for DNA, for semi-conservative replication. Notice, and here am I going to emphasize, always call that double-stranded helix, always call that double-stranded molecule of DNA a molecule, because you can then talk about the two strands that make up the molecule. Never talk about a DNA strand when actually you're meaning the molecule. And don't actually talk about a DNA molecule being made up of two molecules. Yeah, just get your head around that terminology. There is a single DNA molecule is made up of two strands. That's the easiest way to be able to describe the DNA molecule. DNA ligase is one enzyme involved in the replication of DNA. State two other enzymes involved and describe their functions. Can I just say, these are the two enzymes that you have to know about. You may not have been taught that DNA ligase is involved in DNA replication, and you don't need to panic that you haven't been. You will come across DNA ligase in other contexts. In fact, OCR, you do learn about DNA ligase because you learn about the lagging strand. Apologies, yes, you do. But the core content of DNA replication is about these two enzymes. Knowing what they do. I just realised I haven't marked that one. Okay, okay, here they are. And knowing what they do, absolutely crucial. DNA helicase, it breaks the hydrogen bonds between those two strands. And DNA polymerase forms the phosphodiester bonds and it sews up the sugar phosphate backbone. So here is the mark scheme. You had to talk about DNA polymerase because you also about RNA polymerase. You have to say, joins the nucleotides or forms the sugar phosphate backbone. The classic misconception here is this here. This is a classic misconception that DNA polymerase reforms the hydrogen bonds. It does not. It sews up the sugar phosphate backbones of the two strands, phosphodiester bonds. So here I got enzyme 1, function 1, enzyme 2, and function 2. So I got the 4 marks. B, in 1958, Mieselson and Stahl carried out an experiment that provided evidence to support the hypothesis of semi-conservative replication. They grew E. coli on a growth medium that only contained 15N, heavy nitrogen. They then transferred the bacteria to a growth medium that only had 14N, light nitrogen in it. And only then did they allow the bacteria to undergo cell division and therefore DNA replication. After each division, don't ask me how they did this, after each division, the DNA from some of the bacteria was extracted from the culture and centrifuged to separate it. And this shows the bands in the centrifuge tube. So here is the DNA made with only heavy nitrogen. This is the first generation. This is where light nitrogen was the only nitrogen that was made available. So hopefully it makes sense that all the DNA that was made as a result of the first round of replication, the first generation, all of that DNA would have comprised of one heavy nitrogen strand and one light nitrogen strand. Every single molecule of DNA would have been made up of one heavy strand and one light strand. Therefore, there's a single band and it's in the middle of the tube. This is the bottom, this is the top. But of course, because the light nitrogen is only available for these generations, of course, the next generation is only able to make the DNA from light nitrogen. And therefore now you will have two bands in the tube. This one is 14N only, and this one is the DNA made up of 14N and 15N. And as the generations go on, the band of the DNA that is made up of solely 14N gets bigger and bigger. And I'll put a little diagram up in a minute, and I hope it makes sense. So this is what I wrote, and I must admit, 2 marks. I thought this was really hard to describe what was, or to explain, let's be honest, to explain what was going on in a very succinct way. And also trying to work out possibly how on earth the mark scheme wanted you to write it. So this is what I put. Because replicated DNA in generations 1 to 10 can only use 14 nitrogen. So in generation 1, all DNA is made up of half 14N and half 15N, because there is only one band, that's all the DNA, and all of that DNA is made up of half 14N and 15. Sorry, this diagram isn't correct. This is 15N DNA. Oh, sorry, no, I've done that wrong, haven't I? Yeah, sorry, I was right first time. This is this band here is the DNA made up of 14N and 15N. The important thing here is that all the DNA is. In generations 2 and 10, the percentage of DNA made solely of 14N increases. And here is the mark scheme, and I only got 1 mark. Let me just put that in. I only got 1 mark. And I'm hoping that this diagram makes sense. And I've made the 15N red and the 14N blue. Hopefully, if only 14N is allowed here, but you know, to make the first generation or replication of DNA, does it make sense that these two, the two molecules, all the molecules of DNA that are formed, each of them is one heavy and one light. It ends up being a band in the middle of the centrifuge. But notice we only have the original amount of 15N strands, regardless of the number of generations. So does it make sense that proportion increases, the proportion of DNA molecules made up of only 14N increases as the number of generations increase? Do have a look. I've got a video on explaining the measles and style experiment, which I've lifted this from. So I hope that kind of helps. Okay, I think this really is the last question. Protease enzyme bromelain can be extracted from pineapples. Okay, whatever. A student investigated the effect of changing the content of the enzyme and measured the time taken to break down the protein gelatin. So bromelain is obviously a protease. It breaks down gelatin into amino acids, presumably. State 3 variables that the student would need to control in order to make the results valid. Look, notice that it is control variables as well as having the correct methodology that makes things valid. Repetition also does as well. Okay, so these are all the things, you know, what things affect enzyme action. So think of it except for enzyme concentration, temperature, pH, and substrate concentration. Here's the mark scheme. Now this was a rookie error. Got mark 0.1, I got mark 0.2. I didn't get mark 0.4 because I just used the standard term. Oh, substrate concentration. Yeah, so the fact is, and this is absolutely a valid thing that A-level examiners expect, is that you don't just give pat standard answers, but where you can relate a part of your, dare I say it, your pat standard answer to the question itself, you should do so. So here, I should not have used the word substrate because they've told me what the bogging substrate is. Should have said gelatin. Okay, so hope that makes sense. Describe how the rate of reaction was calculated. Right, your big clue about how the rate of reaction is calculated is looking at the units. The units tell you how something is calculated. Meters per second tells you that it is the meters divided by time in seconds, because this -1 says that the set's below the line in the division. Yeah, so this just says seconds to the -1. So it is literally 1 divided by time in seconds. So that's what I put. Okay, so 1 divided by time, or 1 divided by, oh yeah, whatever. Yeah, and you could also put 1 divided by seconds, number of seconds. Explain what the standard deviation shows in table 20. So here am I just wrote my definition of standard deviation. Standard deviation tells you about the spread about the mean. The higher the number, the larger the spread about the mean. Here's the mark scheme. Got the first mark. Yeah, I didn't get the second mark because this question wasn't just what does standard deviation tell you about data, asking you, and I should have picked this up probably, they were asking you about what it tells you in this experiment. So hence the second mark was actually about what's the significance here? What does it tell you? So I didn't get that mark. This shows the results plotted on a graph with the standard deviation as error bars. Explain the pattern in the data using table 26 and figure 26. So I put the table up here. So can I just point out that the size of my writing on screen in relation to the actual lines that are on the screen, my writing size is huge. So hence I do overrun sometimes. So as the bromelain concentration increases, so does the rate of protein digestion. But above a concentration of 0.75%, the rate plateaus. Can I just point out, this is all description. I haven't started explaining yet, but I just kind of, you know, I've got to do some form of description before I can start explaining. But I'm realizing what I'm doing. I'm not expecting, I'm knowing that none of this is going to give me a mark, because the thing is explain, the instruction is to explain. This is because as soon as I write the word because, I'm into explaining. This is because at lower concentrations, more enzyme substrate complexes, you'll notice that this abbreviation is allowed, can be made, but above 0.75%, the concentration of protein is the limiting factor. because it plateaus off. And at concentrations greater than equal to 0.5%, the standard deviation error bars overlap, so the difference in rate is not significant. Here's the mark scheme. Here are those. I'm going to let you look at this at your leisure later. This will all be up on my website, on my YouTube channel immediately afterwards. So I got mark point 1, and I got mark point 2 because I said no. I got mark point 4 because I talked about the substrate. Here's funny. This is funny. They've talked about substrate concentration in their mark scheme, not the protein. The enzyme concentration, bromelain, is the limiting factor. I didn't get any of these mark points because they wanted you to talk about uncertainty, and I just said that it isn't significant. I think here just talked about the significance of it. So this is true, but it isn't the emphasis that was in the mark scheme, at which point I just kind of go, do you know, I couldn't have done any more. I haven't got my code. Oh, and there at the end of the exam, finally. And I hope that has been really useful. I'm hoping that I have, so I'm just going to chat for about 30 seconds now. And unless anybody puts any questions up in the chat, I will sign off until next week when, funnily enough, I plan to be doing paper 2 of the 2017 OCR AS paper. So hopefully that has helped you understand how important it is to read questions carefully, how important it is to notice whether you're being asked to describe or explain, or describe and explain something. And I hope that it has also helped you a little bit with some of the maths, because I do understand that some students find the maths in biology tricky. So I'm hoping that that has been useful. And just from the comments, I just want to say thank you for the thank yous, guys. And I will see you next week for the follow-up paper. Hope you have a good week and see you soon. Many thanks. Bye.